Hello, everybody. Welcome back. So we just finished talking in our last lecture about the plasma membrane of the cells and how that's that double layer to prevent um, things from getting in and out that aren't supposed to. So how do things get in and out then, right? Cells need to take in nutrients and oxygen and things like that, and they need to be able to get rid of waste or to send out things that they make, like in a multicellular organism, to help, that they're making for the other cells. So how do cells get things across that membrane? And that's what we're going to talk about today is crossing the membrane. So here we are looking at the different ways that things can cross the membrane. So when we're looking at this, diffusion and osmosis, these are two words for passive transport. So things that don't require any energy. So some things can go back and forth across that cell membrane without the cell having to do any work. And then some things are too big, or for other reasons, the cell's going to have to use energy to get things either in or out. So those are active transport, and there are a few different types of active transport, and a subset of this is endo and exocytosis here. So we're going to go through these four ways of crossing the membrane. So again, as we talked about already, that membrane is selectively permeable. I don't think we've used that term yet, but that means that some things, select things that meet certain qualifications, can get through it, but other things cannot. So fats, lipids, can pass through more easily because it is that lipid bilayer. Remember, you've got your these what look like little tails or legs here. This is the fat, right? And those are those fatty acid chains, and then there's that phosphate head. So some things can get through easily, some can't. It's selective in what can cross through. Things that can cross through without using any energy on the part of the cell, that is a form of passive transport. So it's transporting things in or out of the cell without using any energy. But if the cell has to use energy to do it, it's called active transport. So we're going to talk about the two types of passive transport. The first type is diffusion, and the second type is osmosis. So again, because this is passive, it happens without using any energy. So a particle, and the particle here is the solute, can dissolve into a solvent. So a solvent could be a gas, or a solvent could be a liquid. So if you're making salt water, salt would be the solute and the water would be the solvent, and it ends up being salty water, right? Or you can make sugar water. And when you do this, that solute is going to move from where there's a high concentration of it to where there's a low concentration of it, so that it's even. Things are going to even out when they're diffusing. So we have a picture here to kind of make it a little easier. If this is your cell layer right here, and we're looking at the passage of time at the as you move from left to right. So on the far left, this picture, you see inside the cell, down here at the bottom, there's none of these little blue dots. And let's say these blue dots are oxygen. Okay, so this is oxygen in your blood. And here's your cell at the and there's all of the oxygen's on the outside of the cell and none of it's on the inside. Things that can pass through that wall are naturally going to pass through from where there's a lot of it to where there's a little of it. Okay, so just to even things out. They're not thinking about it. It's just naturally how things work. This is just physics. They're just going to move around until it's even. Okay, so some as we go through time, like if this is time 1, time 2, time 3, in time 2 you can see a little bit more oxygen has entered that cell, and by time 3 it's an even amount on both sides, inside and out of the cell, equal amounts of oxygen. This is true if you've ever sprayed like an air purifier or perfume or something in a room, right? It starts off very strong where you first sprayed it, and then it kind of evenly distributes itself throughout the room, right? Until you can barely smell it, because it's all these little particles of whatever the perfume is made of, and they're going to be evenly distributed. Or if you put a drop of food coloring into a cup of water, it starts off being very dark, right, with just one drop, and then it will evenly spread out until and it might require a little action like some stirring, right, but it will evenly, even if you don't stir it, eventually just spread out. And so until those little particles of color that are dissolved in the water will be evenly distributed. So if there's this semi-permeable membrane, if this solute is able to go through the membrane, it's just going to be evenly distributed, as if the membrane's not there at all. Okay, it's just going to keep moving until it's even on both sides. So when things are moving across that membrane, they can just move without any help at all, which is simple diffusion. This would be like oxygen, like we were just talking about. This is actually how your cells get oxygen, right? Highly oxygenated blood goes, the blood goes into your lungs. It gets all this oxygen from the lungs into the blood by diffusion, and then the blood gets pumped around your body. And as it pumps around your body, the cells that don't have it take it out of the blood, right? So it goes from lungs to blood and then blood to lungs. This is just simple diffusion. Now, some particles can't just fit through that lipid bilayer, but they can still move through diffusion if they have special doorways to open instead of going through that bilayer. So some large molecules might pass through a transport protein. And I have a picture. I can go backwards. You guys have seen this already, but here, if you can see the one that I'm circling on the far left, this would be a transport protein. So, and it even labels it over here, protein channel. So water could just go through there. It's not going to use any energy. It can't go through that lipid bilayer, right? Because water and fats don't mix, but it has a special gate that it can use to go through, and it will still diffuse, right? So it'll still even out how much water is on one side and water is on the other side. So some things like water will move through those channels. It is still diffusion when they do this. I'm just moving forwards again to where we were. It's still considered diffusion because it's not using any energy, okay? It's just called facilitated. To facilitate is to help along, right? So this is helped diffusion. It's a bigger molecule that can't fit through that membrane, is going to go through like a secret passageway. Okay, but it's still diffusion. So that's one type of passive transport. So there's two types of diffusion, but they're both passive transport, okay? Neither of these requires energy on the part of the cell. Now, if you remember back a few minutes ago, I said there are two different types of passive transport. One is diffusion. The other one is osmosis, which I'm sure you've all heard this term. Osmosis is basically diffusion, but it's when we're talking about water instead of something else, okay? So if it's water that's diffusing, it's osmosis. If it's some other particle that's dissolved in the water or in the air, then it is called diffusion, okay? So as we said, water can't just move through the lipid bilayer, right? So it goes through these special water channels, these transport proteins in the cell membrane. Now water, when you think about which way it's moving, this is where it gets a little confusing for people, because if we take that same picture that we had before, and this is our cell membrane, and we had all these little bits of the solute up here, right? And then and we'll divide it into 3 time frames, right? And this is dissolved in water. Okay, so I'm just going to put a bubble of water around here. This is a ball of water. These little X's that we have here, these actually can't move across. Okay, let's say it's something that can't move across that membrane. But we want it to be even on both sides. If you can't move the other thing, you're going to have to move the water. So normally we had said that the solute would go from where it was an area of high concentration to an area of low concentration if you're moving the X's, right? But if you're moving the water and you want to make it equal on both sides, and you can't move the x's, you just have to add more water to the top one, right? You could either have super saturated, really salty water in a tiny little cup, or you could add another gallon of water to it and it'll seem like there's not really any salt in there, right? You wouldn't be able to taste it anymore. That's basically osmosis. It's going to go from areas of low concentration of this solute, right? So the dissolved molecule here, there's only one of them, very low concentration. The water is going to be moving up. And I'm going to show you a picture to make this a little clearer. Okay, so here we have, here's our semi-permeable membrane. Okay, it's also selectively permeable is the word that we'd used before, so same thing. Selectively permeable, some things can go through, some things can't. You can think of it like a colander when you're pouring your spaghetti in it, right? The water can go through, but the spaghetti can't. The spaghetti can't get out. So here we have all these little purple dots. This is our solute. It's dissolved in the solvent, it's dissolved in the water, and the purple dots can't move. But we want it to be equal concentrations on both sides. So if you can't move the dots, you got to move the water, right? So osmosis would result in all of this water moving from the left side to the right side. Now, if you were, let's say this is salt in the water, if you tasted it in the first picture on the left, this would be super salty, right, and gross, and this would maybe taste like normal, right? Once you move that water over, now they're both going to be the same. They're going to, because the amount of salt per water is equal on both sides. So the water moved from an area of low concentration of salt, and the water moved to where there was a high concentration of salt. So the movement of that water is osmosis, okay? If you've got any questions up to this point, write them down. This is often an area where, you know, things need a little more explanation. We're going to keep going though, and hopefully they make a little more sense as we move through. Okay, so when we're talking about these solutions, right, we had the salty solution and the less salty solution. Isotonic, iso means the same or equal, would be the concentration of molecules is the same on both sides of the membrane. So if we had that same thing here, and you had just a little bit of salt on this side and a similar amount of salt on this side, or at least the concentration was the same per unit water, that would be called isotonic. Okay, if you had one side that had a lot, and here's our water levels, a lot of salt, and one side that only had a little, you would say this one was hypertonic. Hyper means more. If someone's hyperactive, right? They're more active. So this one has more salt. And in comparison, then this one is hypo, which means it has less. But it's always in comparison to each other, right? So if we're looking at a solution, and you're just looking at one solution, you don't know whether it's hypertonic or hypotonic. You have to compare it to something else, right? So if you compare A to B, now you can say which one is hypertonic and which one is hypotonic. Think about that for a sec. Pause it. Okay, come back when you're ready. So A is hyper and B is hypo. But we could compare it to C. That's like saying someone is tall and someone is short. Okay, someone who's 5'5" is tall compared to someone who's 5 foot, right, even. But if someone who's 5'10" comes in, that person who had been tall is now short. So just because A was hyper, in this instance, if we are comparing A to C, suddenly A is hypo and C is hyper. So it's all compared to each other. Is it more or less or the same amount in it? Okay, so once you understand that it's always in comparison to something else, that should help. Now I'm gonna go just for a sec to a a blank file here for us. Okay, so now let's look at this more in terms of like a cell and its environment. So here we have like a beaker of water and we've put a cell in there. Okay. So now, first off, is the cell hypertonic compared to the beaker, or is the cell hypotonic compared to the beaker? Pause it, write it down, and I'm going to write it here too. Okay, so in this instance, the cell is hypertonic. The beaker is hypotonic. Now that you know that, what direction is the water going to move? Where will the water go? Is it going to go into the cell or is it going to go out of the cell? Assuming that those little X's, whatever that solute is, can't move, where the water is going to move. Osmosis is going to take place. Which direction is it going to go in? So pause it, think about it, and then come back. All right, the water is going to move out of the cell. No, I totally lied. That is completely wrong. You say, Professor, you're wrong. The water is going to move into the cell to try to even this out, right? There's way more inside the cell than outside. The stuff can't move, so we're just going to fill that cell with water and dilute it a lot. And that's what's going to happen. So water goes in. The water will always move from low concentration, right? There's a very low concentration of this stuff here. There's none, basically, right? To high. And there's high inside that cell. Okay, let's go back to these files then. Okay, so we're just going to stop here with this last cell, just showing you how what helps plants kind of stay up, right? Have you ever wondered, like, how does a plant, like a stem or like a leaf of grass, stand up straight? And it is because of how they keep their cell walls. So they actually keep all this water. If we look on this one on the right, inside their cell, like in a vacuole. So it's like an extra packet inside the plant cell, and it makes it extra full. Like if you have a water balloon that you overfill, it's going to be, it's not going to flop over, right? It's going to stand up straight. You can have a pretty rigid balloon if you overfill it. If you forget to water your plants, right, they're going to start to droop. That's because they can no longer keep all that water in their cells, and that's when they start to wilt. Okay, so we'll stop here, and we will finish the talking about active transport, right? This was our passive. We'll get into active with the next lecture. Bye-bye.